Today we'll talk about the fundamental tissue and mitosis. We will have three parts of this. The first part has to do with a virtual microscope, how to identify things from the images by using the computer, describe the steps in tissue processing, and discuss the different types of microscopy, both light and electron microscopy. Then we will interpret cytologic features of cells by different staining, differential staining, with hematoxylin and eosin are main stain, but we also use PAS and toluidine blue, as well as processing tissue for conventional transmission electron microscopy as well as scanning electron microscopy. Part 2 has to do with recognizing and identifying cells using the microscopic preparations that we have, the images that we have, a little bit about how to observe the different structures in the images that we have, and to understand the relationship between structure and function of cells. Part 3 has to do with mitosis, talk about the stages of mitosis and cellular structures that are associated with mitosis. Mitosis is a process of cell division by which you produce two daughter cells identical to the previous parent cell. Enjoy. Fundamental tissues and mitosis. This is medical school histology. Part 1 will talk about the types of microscopy, tissue preparation, and staining. Part 1 we see the virtual microscope. In other words, we're looking at images of things on the computer and not really using a microscope per se. The microscope was used to take those images. Describes the steps in tissue processing and discuss different types of microscopy. Interpretation of cytologic significance of differential staining by hematoxylin, which stains the nuclei, and eosin, which stains the cytoplasm. Part 2 and 3 will be composed of other components. The virtual image. Here we see the images that we're looking at. Here we see a low view of what we're looking at. If you wanted to click here, it would go to there. If we wanted to click there, it will go into this region, and it shows you, a little box shows you what you're viewing. So here we're viewing what's in this box right through there, and as we change the magnification by rolling your thing, you can see the box is getting smaller. We can move the box over to here, and the images will come in. Here we see some mucus secreting glands, and then this is skeletal muscle cells. You can see the blood vessels in between. Let's see if I can get one that's good in focus. As we can see there, you can see the little capillaries between the muscle cells. The muscle cells, these are the nuclei of the cells. This is actually artifact here, the space that is in through there. You see I'm just changing the magnification. Here we see more skeletal muscle cells, long cells, more mucus secretion. These are white fat cells. These are fat cells. This will be one cell itself, and this is a fat globule that we see inside there. Here we see epithelium. This is stratified squamous epithelium associated with the little projections in the tongue. So this is the epithelium on the surface, and then the connective tissue below there, and then of course we have muscle below that, and then we can see different glandular structures in through there. Actually, this is artery and venule that we see in through there. These are the glands, and we can see ducts that carrying the glandular secretions out to the surface. Also, we can click on here and it will show you the magnification. You can change the magnification right here too, but by clicking in here you can also, so you can move this back and forth, or you can move over here and look at this blood vessel. See, this is our artery right here. This is smooth muscle of the artery. These are the mucus secreting cells with a flat nucleus in the surface, and you can see again, if you look over on the right hand side over here, you'll be able to see the columnar type cells that we see there, and these little capillaries associated with them. If you want to look at a different piece of tissue, come over here and click. on that one, and we can see it's a different tissue. Here we can see nerve. So this is perineurium in through there and around there, and these are myelinated axons. The axon is dark, the myelin sheath has been removed. So this is nerve, skeletal muscle cells, more nerve, nerve in through there. And we see this is a artery or arteriole, you would say, kind of small artery. Tissue's been cut at that side. We want to click over here. We can see stratified squamous epithelium on the surface with projections in through there. Don't really see any taste buds per se. There should be some taste buds along here on the sides. I don't really see them though. But you can see blood vessels, muscle. These are different ways you're able to visualize the image. Change the magnification, move this back and forth to change the image or to observe different images if you want to. Now the microscope that we'll be using the images of mainly is a light microscope, and as a rule we use bright field, which have stained dead specimens that we are observing. We have our eye will be looking through the ocular lens. There's a prism in here. These are the objectives that will move around to change the magnification. We have the specimen slide that's on there sitting on the stage. Below the stage we have a condenser which condenses the light going onto the specimen so you're able to view this with your eye. The light source coming through here. Here's the focus knobs. The coarse focus in the middle and the fine focus on the outside, and then there is the bulb. Now whenever you're using a light microscope, there are some features that should incorporate a color illumination, and it has 5 different steps. First of all, you put the specimen on there and then you focus the specimen. Here's the coarse focus and the fine focus. You focus the specimen once it's on the slide. Then you close the field diaphragm, which limits the light going in through there, and then you focus the condenser. So here's where the condenser knob is focused right here. This is a condenser, but this is a focus knob for the condenser, and you're actually focusing on the field diaphragm that you've closed. The next thing is a center, and here we see the centering knob of the condenser. So you're centering that little light spot that you see. And then the last step is to open the field diaphragm. So if you use color illumination, this procedure, each time you use a microscope, it will reduce the fatigue associated with it. There are different types of microscopy. The light microscopy, the bright field, the most common, they use with stained slides, dead slides. Phase contrast is a type of microscopy that lets you observe living cells. So a characteristic thing you'd like to see with living cells is maybe its cell cycle, mitosis, that we'll be talking about later on. Nomarski differential interference contrast microscopy is another type. It allows you to optically section through the specimen, but it looks like a lunar shot, something from the moon. Dark field microscopy is another type of light microscopy. All these are light microscopy. Then we have transmission electron microscopy, which magnifies the images and allows you to visualize intercellular structures. The wavelength of electrons is much smaller than white light, and as a consequence the resolution is much better with an electron microscope. Another type is a scanning electron microscopy, which allows you to see surface images of the tissues. Here we see bright field, and this is usually use it with stained dead cells, and this is not stained, so it shows you that if you don't stain the tissue, you can't see it very well with bright field microscopy. In contrast, phase contrast, you can see several organelles, different structures, the nucleus inside the cell. Nomarski differential interference contrast allows you to optically section up through the tissue. As you're viewing, it looks like a moonshot, and then the dark field microscopy has a dark background with a light for the specimen that you're seeing. So here we see the nucleus and the cytoplasm, nucleus, cytoplasm, nucleus, cytoplasm, nucleus, cytoplasm, as we can see. Now, there are different types of electron microscopy. The conventional type of electron microscopy is where you have electrons coming down through the specimens, and they hit a screen on below, and it's kind of like X-ray. You see a negative component, but this is actually a positive view of the negative image that you see. So this is your conventional electron microscopy. There's also a scanning electron microscopy that lets you see the surface images of things. And then there's also a carbon replica transmission electron microscopy, where here and here we actually, with conventional microscopy, you actually have the tissue there. In this case, it's only the replica. So you're only looking at a replica of the specimen is what we see. So we have a conventional carbon replica scanning electron microscopy. And here we can see the typical conventional electron microscopy. This is a rough endoplasmic reticulum, mitochondria, nucleus, centrioles. You can see the microtubules associated with the chromosome pulling apart, centriole, some of the organelles you can see. That's conventional. This is scanning electron microscopy that you see this blood vessel. You see there internal elastic lamina very nicely. You see the surface view of images, and this is in the kidney. This is one of the renal corpuscles. These are podocytes on the surface, little foot processes surround the capillaries, as we'll study later on. Now, whenever you're looking at transmission electron microscopy, you want to compare magnifications. Be mindful of changes in magnification. And a good thing to look at for the magnification is a mitochondria. How big are mitochondria? Here you can see at this 9,000 electron microscopic magnification, you can see mitochondria in these little lymphocytes. These are mitochondria here and here. In contrast, we can see mitochondria there and there in this cell. Here they're getting bigger. We can actually see their cristae there, and they've got more than one membrane, and an 80,000 assembler type thing. Ribosomes, you can hardly see ribosomes. You see the granularity there. A little bit of ribosomes might be able to be seen in through here, this precursor to a red blood cell. See ribosomes here, but here we can see ribosomes at a higher magnification. Here we can see plainly, we can see ribosomes on the rough endoplasmic reticulum. If we look at a little higher magnification, 60,000, you can see ribosomes very nicely in through there. There's polyribosomes, groups of ribosomes here, and here even see that there's actually a couple of units in each of these little ribosomes that we see. Also at higher magnification, this is 200,000, we can see that the membrane that are running here and here is really like a railroad track. It has a trilaminar structure, a light in the middle and two dark sides on the outside, which is part of the membrane structure, the lipid bilayer that we see there. Now, in terms of preparation of tissue, first of all, you have to fix it. And you have some chemical fixing, formaldehyde. That's kind of like what you embalming bodies. You fix them. You dehydrate it with organic salts, usually alcohol start with, and then you embed it in a paraffin or plastic, and that let it stiffens so you can then cut the tissue. Before that time, you can't cut the tissue. It's kind of like boiling an egg, and you can really thin section the egg if it's boiled hard, and that's the purpose of the embedding. Sectioning is actually the cutting itself, and it's from 1 to 10 micrometers in thickness. Staining is where you have different stains, and the stains stain different biochemical components of the cells that allow you to see different structures in the cell. And then the mounting on the slide, and then you histologic observing with a microscope, and that's been done for the bulk of the specimens that we'll be looking at, because we'll be looking at the images thereof. So again, you fix it to preserve it, prevent it from rotting. You embed it to make it rigid with paraffin or plastic, and then you section it with different types. In electron microscopy, it's very thin sectioning. And so here we see you fix it in Bouwens or xylene or Zinker's solution, or formaldehyde or glutaraldehyde, I should say. And then that preserves the morphology. Dehydrate it to clear it, clearing to make the embedding media be able to go inside the tissue to make it harder. You can see this takes 12 hours, 24 hours, 1 to 6 hours. A fair amount of time to process tissue. Once the tissue is inside the block, the paraffin block or the plastic block. Then you make sections. So this microtome is going to come down. As it comes down, it's going to cut sections, and then it advances 1 to 10 micrometers each time, and so that's the thickness of each of these sections. You take these sections, mount them on a slide, and then stain them, and then observe them with your microscope. And we can see there are different types of stain. We got hematoxin and the eosin are most common, and that's what we see here and here. Hematoxin is a blue-black dye for basic pH. So in other words, it binds nucleic acids. In DNA is what it does. Ribosomes as well, which have nucleic acid. And here we see dark blue nuclei of lymphocytes. These are the nuclei of lymphocytes. In contrast, eosin dye is a red dye, is acid dye, and it stains proteins. So here we see the protein staining of the smooth muscle cells. Hematoxin eosin of the kidney. We can see the individual cells here. Here's a nucleus, and the nucleolus is a dark structure inside there. And then we can see the basement membrane around the cell. This is a corpuscle here with glomerulus inside the blood vessel. Also, this is another kidney. Another type of staining is the periodic acid shift, and that allows you to observe the location of carbohydrates. And that hot pink that we see here and here shows you the brush border. So this is a microvilli, uniform microvilli on the surface of the cells that have a glycocalyx coat. It's a glycocalyx coat which contains a sugar and is PAS positive, they would say. Also, epithelium, which is what this is, sits on a basement membrane, and that too is PAS positive, and you can see the PAS staining of that. So in light microscopy, we have hematoxin and eosin. This is a kidney again. You can see the Brattle cells, Bowman space, and periodic acid shift. Here we can see the basement membrane as well as the brush border that you see in the proximal tubule. Also, carbohydrate rich is the basement membrane of the capillaries as well as the podocytes. That's what we're seeing in through there. It's counterstain for the nuclei. So those are the nuclei. Here, the cytoplasm. And note that this duct is different from that. These are proximal tubules here and here, but the pink, without the pink, is a distal tubule, proximal distal tubule. And you can see that by the brush border that's located in the surface. Another way they might see some is toluidine blue, where everything is blue. And in fact, whenever you look at H&E and PAS, you look for color clues. Colors can tell you the light or dark or what color it is. But in toluidine blue, everything is blue. So you have to look for the shape, size, and staining intensity to give you some clues about toluidine blue. So here's hematoxin, but also this is a staining, lead citrate staining for electron microscopy. So this is the glomerulus up there, and here we can actually see this is the capillary and endothelial cell through there, but these are podocytes. So this cell is the same as that cell, using two different types of microscopy. You can see how the details that we can see at the electron microscopy that we cannot see up through there. Here's a little capillary. You see the fenestrated, and there's a capillary here, but we can't see the fenestra at that magnification. Also, there's artifacts, and one you need to guard against. These are man-made distortions or in the tissue. So these are not real structures. And here you can see in the kidney again, where this transitional epithelium has pulled off from this tissue here. You can see it's just free in there. Tissue is just not free in the lumen. It's usually attached to things. And this is an odd staining as well. So you can see a higher mag of that. So it's kind of got pulled away. You can see the nice cells there, but it is pulled away, and that's an artifact. Artifact is a man-made distortion, not real, and you have to guard against them. So identifying cells, you've got the cell, which is a basic component of life. Your tissues is more than one type of cell, more than one of a type, a group of cells. Epithelium would be a tissue. Mother wound tissue makes organs, organ system, and then you make a whole human. Cells are two types. There's somatic cells, those are muscle cells, and then there's germ cells. Germ cells are they're gametes. They're either going to make sperm or going to make egg. Those are the germ cells. And there's morphologic characteristics of certain functions of cells. Sometimes you have lysosomes, sometimes you have structures inside there that's being secreted. The nucleus, nuclear pores, rough endoplasmic reticulum, the Golgi apparatus, we see there, Golgi apparatus, centrioles, mitochondria, cilia, with the basal bodies here and cilia projecting up, and then these are microvilli, which have microfilaments inside there. So those are some of the organelles that we want to be able to identify. Thank you.